going to talk about the Endosymbiotic Hypothesis. So let's spell that out. Endo, which means to engulf or to bring in. All right, and this is a hypothesis. This is not something that's been proven. It's just something that we believe to be true. So the idea is there was once a very large prokaryote, and it essentially swallowed a smaller one. So there was a smaller prokaryote, and it got engulfed, and you might say eaten, by the larger one. Now there are two processes that we are going to learn very soon in upcoming videos called Endo and Exocytosis. Alright, and Endo and Exocytosis are the process of the cell bringing in large things or large groups of things into the cell all at once, or expelling them all at once. And so the idea is there are these processes, not just like pores or pumps that pump small amounts in. Endo and exocytosis, all right, they bring in large amounts all at one time. Now once this small cell was inside the larger one, it did not die, all right? In fact, it thrived, and it became what we now know as the mitochondria. We also think that the chloroplast in plants also originated in this method. If you don't remember what a chloroplast is, it's the thing that does photosynthesis. It absorbs energy from the sun and provides it to the plant. In our cells, we have mitochondria. Plants have both of these, and you may hear the term plastid. Plastid is referring to both of these. Both of these are plastids. And these are organelles. And now we've talked about organelles, and organelles are compartments within the cell where you do specialized chemical processes. And these organelles, they're like organs are to our body. These are the equivalent for the cell. The cell does specialized processes there. And the mitochondria and the chloroplast specialize in producing energy for the cell. For us, we use mitochondria to produce ATP, and that ATP, of course, is what powers our cells. It allows the proteins to change shape, and it powers pumps to pump things out and in. And eventually we'll see that there are all types of things that ATP does for you, and that the organelles, all right, are constantly moving things around, taking things out or bringing them into the cell, that sort of thing. And we talked about the endo and exocytosis, and we'll talk more about it. But it's believed that at one time this was the process, the endo and exocytosis, that brought these smaller cells into a larger prokaryotic cell. And we believe that this cell eventually evolved into what we now call eukaryotic cells. In other words, a cell with a nucleus, all right, with chromosomes in it. And another reason that we think that these little mitochondria, let's draw them a little smaller compared to the nucleus, it's going to be tiny. All right, well, these little mitochondria have their own DNA. Now the mitochondria, let's see here, let's find some space here. It has a double membrane, but inside the actual mitochondria is a little loop of DNA that reminds you of a bacterial DNA. It's not like the chromosomes in a eukaryote. So that's another reason that we believe that the mitochondria, all right, it's much smaller here, blown up there. We believe that it was once its own organism. It was once a smaller prokaryote. And the genes inside remind us of prokaryotes. And in fact, they have quite a few statistical properties that in common with bacteria. And not only that, we have found several genes up here that are regulated like bacteria. They're what are called operons, and humans actually have 4 of them, and they behave like bacterial genes, and they are statistically similar, and they're missing from the mitochondria. So the mitochondria has lots of genes that remind us of bacteria, but they're missing the ones that we find up here. Now they're missing a lot more besides those 4, but those 4 are found here, and they're not found in the chloroplast or the mitochondria, and therefore we believe that they migrated to the nucleus. They were safer there, and the others remain in the chloroplast and the mitochondria. And so they are sort of independent in that they have their own DNA, but they absolutely cannot live outside of the cell. They would die if they left. So they're not like regular bacteria that can live just anywhere. They're sort of a dummied down, stripped down base model, even less than that really. They absolutely cannot live outside of the larger eukaryotic cell. But they do have their own DNA, and they do remind us of bacteria in several ways. So understand the endosymbiotic hypothesis. Understand it is only a hypothesis. It is not something that we have been able to prove. We have not seen a cell do something like this, and we cannot make a cell do this. So we don't know exactly how it happened, but we believe it happened sometime in the past. And we believe that there are other organelles, but that are similar. There are mitochondria and chloroplasts. There are things that can remind you of a mitochondria and chloroplast, but they're not similar enough for us to think that they were once living organisms. And we're going to learn about the rest of the organelles, and we're going to learn about the endo and exocytosis. But I thought for this test it would be a good idea to start you off with an interesting story that would kind of set up all the other things that we're going to learn. So we're going to learn endo and exocytosis, we're going to learn about pumps and pores, we're going to learn about the organelles. So all of these things are related to the endosymbiotic hypothesis, and I absolutely will ask you a question about this on the test.